For decades, the biological sciences operated under a strict, unifying framework. According to this central dogma, the instructions for all life and all biological processes had to be written in nucleic acids, DNA or RNA. This rule governed how we understood infectious disease. Bacteria, viruses, fungi, and parasites all contain genetic material. They use these blueprints to survive, multiply, and spread from host to host. The consensus was absolute. Without DNA or RNA, an organism could not replicate. A pathogen devoid of genetic material was considered a biological impossibility. Any evidence to the contrary would require an entirely new mechanism for how life and disease could propagate. In the fall of 1972, a resident neurologist at the University of California, San Francisco, named Stanley B. Prusiner, encountered a patient suffering from progressive memory loss and declining motor function. She was diagnosed with Creutzfeldt-Jakob disease, or CJD. The pathology of CJD is relentless. It systematically destroys the patient's brain over a matter of months. Yet, strikingly, it provokes absolutely no response from the body's immune system. There is no fever, no elevation of white blood cells, and no antibody response. At the time, medical literature classified the cause of CJD and similar diseases as a slow virus. But existing data from Tikva Alper showed the infectious agent was extremely resistant to radiation. Since radiation destroys nucleic acids, this strongly suggested the agent contained no DNA or RNA. The literature filled with conflicting hypotheses attempting to explain this. Prusiner realized the implications of this medical mystery. He was looking at an infectious killer that appeared to reproduce and spread without the genetic material required by every known law of biology. Prusiner spent the next decade attempting to isolate this elusive pathogen. His work accelerated significantly when the laboratory developed a bioassay using Syrian hamsters, which exhibited a shorter incubation time, allowing the team to purify the infectious factions. The results were clear. The agent was resilient to procedures that altered nucleic acids, but deactivated by techniques destroying proteins. The pathogen was a protein. In 1982, Prusiner introduced a new term for this infectious particle, the prion. The publication provoked intense skepticism. Virologists argued fiercely that a protein without genetic instructions could not possibly cause an infection. The concept was labeled a scientific heresy. To defend his data against a highly critical establishment, Prusiner had to demonstrate exactly how a solitary, lifeless protein could replicate in the brain and spread from host to host like a conventional virus. The next major breakthrough came in 1984 when researchers isolated the gene that encoded the prion protein. They discovered that this gene wasn't a foreign invader. It was naturally present in the DNA of all mammals, including humans. Every day, our bodies produce the normal cellular form of this protein, PRPC. In its benign state, it folds into smooth, coiled alpha helices. But it has a vulnerability. It can misfold, flattening into rigid beta sheets. This abnormal conformation, PRPSC, is highly stable and indestructible. When this rogue protein encounters a normal alpha helix, it acts as a physical template, forcing the normal one to unfold and adopt the same jagged shape. One misfolded protein creates a second, two create four, setting off an exponential cascade. Through this geometric coercion, the protein spreads its structural defect without a single strand of DNA or RNA. The shape itself is the infectious agent. As misfolded proteins multiply, they bind into dense aggregates the body cannot break down. These accumulations destroy neurons, leaving the brain tissue riddled with microscopic, spongy vacuoles. This single misfolding mechanism provided the explanation for a spectrum of fatal neurological disorders. It is the underlying cause of bovine spongiform encephalopathy, widely known as mad cow disease, which devastated cattle populations and agricultural economies. It also explained Kuru, a localized epidemic among the Fore people of New Guinea, where the infectious prion proteins were transmitted laterally through ritualistic cannibalism. Diseases in sheep, cattle, and humans, once thought to be unrelated, were all tied together by Prusiner's data. A biological anomaly unified a global class of fatal brain diseases. Conclusive proof arrived in 1992. Researchers engineered transgenic knockout mice that completely lacked the gene responsible for producing normal prion proteins. When these mice were exposed to heavy doses of infectious prions, they remained perfectly healthy. Without normal proteins to corrupt, the chain reaction could not occur. Concurrently, researchers around the world who had spent decades searching for a viral genome came up empty. The evidence for the prion was overwhelming, and the scientific establishment ultimately accepted the data. In 1997, Stanley Prusiner was awarded the Nobel Prize in Physiology or Medicine for the discovery of prions. The ruling verified a new biological principle of infection. Alongside bacteria, viruses, fungi, and parasites, science recognized an entirely distinct class of pathogen. The implications of Prusiner's work extend well past the specific diseases he initially studied. The core mechanism he uncovered, protein conformational change, now provides the theoretical foundation for investigating more common forms of dementia. Evidence strongly suggests that similar processes of misfolding and aggregation drive Alzheimer's and Parkinson's diseases. Today, as the director of the Institute for Neurodegenerative Diseases at UCSF, Prusiner continues to build on this foundation, utilizing prion biology to develop pharmacological strategies for the prevention and treatment of these complex disorders. By trusting the physical evidence in front of him, Prusiner proved that biological laws are written by observation, not by consensus.